Okay, welcome to MINEPSYC Part 2. I'm doing it a little bit different this time because last time I struggled really hard with explaining concepts while playing Minecraft, and now it's gonna be a voiceover. So we have the nervous system. Today we're gonna discuss the nervous system, the brain, parts of the brain, how to understand the brain, like MRIs and CT scans and stuff. And lastly, we're gonna talk about how axons, neurons, and those stuff work. The nervous system consists of two parts, the central nervous system and the brain. Central nervous system and the peripheral nervous system. Central nervous system has two parts to it, which is the brain and the spine. The brain is the core of the nervous system, it's the actual part that works, while the spine is is the nerves that put the nervous system to work. So you have the outer part of the spine, which sends messages, and the inner part of the spine, which is the part that saves the lives, which is what do you call this? It's the part that has the life fight. It's like when you touch something really hot, you pull your hand out of it immediately. That's where the spine works. And there's three parts to the spine, which is there's three types of neurons in the spine, which is afferent, efferent, and interneurons. Afferent with an A sends senses from our body to the spinal cord to the brain, while efferent with an E sends from the spinal cord to our muscles, and interneurons is afferent to afferent. So A to E, which is for our fast movement and our like stuff like that. The peripheral nervous system has two parts as well, somatic and automatic. Autonomic. Somatic is our motors, how our how we function, like our motor, hands, movement, feeling, those stuff. Well, autonomic is the automatic stuff that you don't even think about. And drink some water because I keep coughing. Okay, next. We have glands. And our glands are pretty much how we get hormones. So we have pineal gland, which is for melatonin, which is how we sleep and regulate that stuff. Thyroid is our growth and our metabolism. Pancreas is for insulin. Gonads are for our ovaries and our testicles. And adrenal is for our adrenaline. It's stuff that go out and relieve our stress when we are in stressful situations. And all of that is controlled by our pituitary gland. Next, we have neurons. Neurons, neurons are neurons are the building blocks of our nervous system, and a bundle of neurons is called a nerve. Neurons and form tissue called neuron tissue, where tissues are stuff in our body that has a distinct function. So neurons, they just send signals all over our body. It was discovered in 1879 by Santiago Cajal, and he theorized the nervous system. The nervous system so our neurons have dendrites, which receive information from other cells, soma, which is the actual cell body and everything that the cell needs to live, axon, which is the really long line that the neuron has, and the axon terminal, which is at the end, and it's the one that sends information to other neurons. So we have these things that wrap around axons. They're called myelin sheets. They're produced by glial cells. And these myelin sheets produced by glial cells, they insulate and protect these nerves and our neurons. And they also improve and make it faster. Make the sending of information much faster. Now for glial cells. Glial cells hold neurons in place. They're not neurons, but they're near the neurons and they are attached to it. And what they do is they supply oxygen and nutrients to the neurons. They insulate the neurons from other neurons and they destroy the dead neurons. They like eat it and decompose it. So there's two types. There's oligodendrocytes and Schwann. Oligodendrocytes are in the central nervous system, and Schwanns are in the peripheral nervous system. And they're the ones that coat the neurons with myelin. And a thing about it is that Schwanns, which is in the peripheral nervous system, can regenerate, while they can regenerate damaged fibers, while the one in the central nervous system cannot regenerate. And from that, now we know what the parts of a neuron. Now we have to understand how it works. There's this thing called the neural impulse. The neural impulse is how it sends information. So it starts from rest. When it's in rest, then the inside of the cell is positively is negatively charged, while the outside of the cell is positively charged. This is because there's outside of the cell, there's a lot of Na and Cl particles, and the abundance of Na makes it positive, and inside the cell there's positive potassium and negative protein, and there's an abundance of negative protein. So what happens is, from a stimulus, the stimulus could be anything. It could be touch or whatever, or feeling something. From those stimulus, the or it can be also be from the brain. These stimulus sends sodium through to the dendrites, and from the dendrites it goes to the axon. Axon goes to the nodes and goes all the way to the axon terminal, and that triggers because sodium channels open because of the semipermeable membrane of a soma, and it enters the the negative inside becomes positive because of all of the sodium coming in, while the outside has all the chlorine. Sodium enters, potassium also everything enters, becomes positive. And then after it gets all of those, after it reaches the threshold and jumps up, it goes back down because the channels are opened up again, and it tries to balance itself up as fast as possible so that it can be prepared for the next time it's used, which is probably in a couple milliseconds. So and there's this thing called a hyperpolarization, where instead of just being negative 70 when it comes back, negative 70 millivolts in charge, negative 55 millivolts in charge, it jumps to negative 70, and then it balances out back to negative 55 millivolts. Next, there's this thing called neurotransmitters. These are the actual stuff that the axon terminals send out. These neurotransmitters they shoot out of the axon terminals, they go to the dendrites, and then the dendrites feels it, and it goes back. So, this is what happens. From the axon terminals, goes through something called a synaptic gap. The synaptic gap is the fluid in between dendrites and the other neuron behind it. And the neuron behind it, which has axon terminals, sends these stuff called neurotransmitters. And neurotransmitters are these. They are ACH. ACH just sends regular signals. Just signals just saying, okay, move, do this. It's for muscle action and learning and memory. And there's these ones called dopamine. Dopamine is for pleasure. Too little dopamine causes Parkinson's, and too much dopamine makes you schizophrenic. Serotonin controls the sleep and mood, appetite, and if there's low levels of it, you get depressed. Glutamate is for learning and memory, and it's for changing the brain's connection, and it can change the brain's connections among neurons, and excess of it can cause stroke, Alzheimer's, and Huntington's. And GABA is the stuff that makes us calm. GABA is also part of a fun fact about GABA is that alcohol increases the effects of GABA, and alcohol is something that's called an agonist. That's why alcohol makes people calm. Next are endorphins. Endorphins are like morphine, they're body painkillers. People get addicted to heroin because their body doesn't produce endomorphines when they get addicted to heroin, so they can't get out of it. And then there's norepinephrine, norepinephrine, which is for arousal and when we are under stress. There's these things called agonists and antagonists. Agonist is stuff that reduce neurotransmitters, and antagonists reduce neurotransmitters, and agonists increases the effect of neurotransmitters. And I forgot about this part. The way that these neurotransmitters come back to its home, which is the axon terminal, is by either diffusion, reuptake, or enzyme degradation. Diffusion is when it just drifts away, coming out of the dendrites. Reuptake is when it returns to the axon terminal. And enzyme degradation is when the dendrites release an enzyme to degrade ACH. Only ACH. Next, when we're gonna study the brain, there's ways to study the brain, many ways to study the brain. There is lesioning studies. There are lesioning studies and brain stimulation. Lesioning studies is when we deliberately break a part of the brain, and brain stimulation is when it's just temporary. In lesioning studies, you get a wire with an electrode, you put it in the brain, and then you just kill it, and then you see what happens. While brain stimulation is just temporary, it's mild, there's no damage, it's not forever. There's two types of brain stimulations. There is deep brain and optogenics, where deep brain is when we get an electrode wire and we put it inside the brain, and optogenics is we just want to see what's inside, what's happening. And there's also non-invasive, where it's literally just outside the head. And transmagnetic simulation. Mental simulation, scalp. Okay, next. There's image tech, the way that we image the brain. So, when you think of imaging the brain, you only really think of two stuff most of the time. It's just CT scans and MRIs. And that's kind of correct, but there's other ways of doing it. When you want to see what's inside the brain, use a CT scan or an MRI. A CT scan uses radiation and X-rays to see parts of the brain, but it's very blurry, and it uses radiation, so pregnant people can't use it. MRI, on the other hand, uses magic, radio waves, and magnets to see parts of the brain. You can see it very clearly, but it cannot have metal. Like no metal, no magnetic metals, or else it's gonna burn or it's gonna hurt. Like people who have iron fighting tattoos, that's gonna hurt like hell. And then that's for seeing it. But to see the function, what the functions of the brain are, and which part function when you do this thing or that thing, you go to NIRs, and fNIR. And NIRS, it's just light. You can see how blood flows inside the brain, but it's very shallow how you can see it. While fMRI, you can see the blood flow deeper, but you have to be completely still and it takes a while. But for real time, to see what happens real time, for example, you want to see how this person's brain reacts when they do this thing, and not how this person brain reacts when they're under a machine. Then you go to EEGs and ERPs. EEGs and MEGs, I mean. EEGs are to see the brain activity through electricity. So it's like you put a helmet and then it sees the brain activity real time. And MEGs also put a helmet, but it uses weak magnets instead of getting the electricity waves. Next, parts of the brain. The brain has many wrinkles called Riri and Sulky. Fun names. Thing about the brain is that it's backwards. The left side of the brain controls the right side of the body, and the right side of the brain controls the left side of the body. And it's connected by something called the corpus callosum. Okay. There's three main parts of the brain. which is the forebrain, the midbrain, and the hindbrain. The forebrain controls emotion, thought, and memory. Midbrain controls sensory motor, reflex, vision, and hearing. The hindbrain controls breathing, heart rate, coordination. It's like the further out you get from the center of the brain, the more complex it is because the hindbrain is in the super center, around it is the midbrain, and around that is the forebrain. The way to get it is that the hindbrain is what we need to live. It's like how a reptile. It's like reptiles. A midbrain, we have reflexes, vision, and hearing, and how we process information. So it's like a dog. And forebrain has emotions, memory, thought, and actual processing. So inside the hindbrain, there are the parts, which is the medulla. The medulla functions, and it's the part that actually regulates our body, how we breathe, how we heart rate, how we swallow. It's our functions to live. And there's this thing called the pons. The pons decides which side of the body signals from the brain relays to. So if the left side of my brain is thinking, then it relays the information to the right side of my body. If the right side of my brain is thinking, then it relays information to the left side of my body. And it also controls our dreams and our sleep. That's pons. The reticular formation, this is for our alertness, for our thinking. It selects what information we remember and selects no, it selects the information we focus on. So, for example, you're driving, you focus on driving, but you don't focus on the sounds that are happening inside the car. You don't focus on your feet itching. You don't think about how the aircon is blowing into your face. You don't think about that. You don't even see the glasses on your face. That is what the reticular formation is for. And the cerebellum controls involuntary muscle movement, like stuff that you're already used to. For example, riding a bike, playing an instrument, typing on a keyboard. It's your muscle memory. That's the cerebellum. And now for the limbic system. The limbic system is part of the forebrain. You have the thalamus, which controls the lobe, which has the lobe where info goes into. So you get stuff from your body, it goes to the thalamus, and it says which lobe of the brain will this information from the sensory of my body go into. And then you have the hypothalamus, which controls hunger, thirst, sleep, our sexual desire, our body temperature, our emotions, and our desires. And then we have the hippocampus, which filters information and decides which one we remember and which one we don't remember for the long term. And the hippocampus only works when we are asleep. That's why sleep is very important because that's how we remember long-term information. And then we have the amygdala. The amygdala is our response to fear. It's a defense mechanism made by our brain from our prehistoric era. Because when we are afraid, it's type of defense mechanism. Yeah. Then you have the cingulate cortex, which is for our emotion and our cognitive processes, and the olfactory bulb, which is right behind our nose and it processes smell and processes stuff on our smell. And the reason why we have a whole part of the brain just for our smell, because smell is a very important thing. It's how we taste things, how we know danger. Like before things even get near, you already get to smell it. And then now we move on to the cortex. The cortex is the wrinkly part of the brain. It's this part that you see outside. And the cortex has cerebral hemispheres. And the cerebral hemispheres is part of cerebral hemisphere has no the cortex is the wrinkly part of the brain, is what you see, and it consists of 5 parts: the cerebral hemispheres, occipital lobe, parietal lobes, the triumphal lobe, and the frontal lobe. They're the lobes. That's what the cortex are. And the cerebral hemisphere is the cerebrum, which is the upper part of the brain. It's covered by the cortex, and it's it's linked with the corpus callosum. And you have the occipital lobe, which is occipital lobe, yeah, which is for visual. It's for what we see. It's a lobe for that. And we have parietal lobes, which is located the front and the back of the brain. It's how we feel in our senses, like how we feel our touch, or how hot it is outside or how cold it is, what we taste, and how our body is positioned, like if we're upside down, leaning forward, or whatever. And you have the temporal lobe, which which is for auditory stuff. It's located right beside our ears, and it's how we hear things and process our hearing. And the left lobe is the one for language function. And we have our frontal lobe. Our frontal lobe is the one that is actually for thinking. It's where we store long-term memory. It's our personality. It's our speech. It's our motor movement. And there are association areas, like areas part of the brain where we associate these parts too. Broca's area is for speech production, and the wrinkles area is for understanding what we say and the stuff that other people say. Okay, that's it. Thank you for watching MindPsych episode 2. I'm gonna sleep now. Good night.